Alrighty, everyone, we'll go ahead and get started. So welcome to the Black and Neuro Seminar Series. My name is Ashley Cunningham, and today we're excited to have and we're excited to have Kellie Williford today to talk about her journey in grad school and navigating pregnancy. And I wanted to point out that there is a live transcript option down at the bottom of your screen, so you can just click the CC button if that's something that you need. So I'm excited to welcome Kellie Williford, and Kellie is currently a Ph.D. student at Vanderbilt University in the Windsor Lab, where she has a F99 DSPAN award and is a former recipient of the HHMI Gilliam Fellowship. Her thesis work is focused on understanding the cell type specificity and circuit mechanisms underlying stress processing and development anxiety-like behaviors. And she plans on applying her knowledge to the field of addiction research as a postdoctoral fellow and beyond. She graduated from Washington University in St. Louis and was a member of the MARC uSTAR research program. So with that being said, we're excited to welcome Kellie Williford. Thank you.
All right. Hello, everyone. I'm honored to be here presenting for the Black and Neuro Seminar Series, and I'm excited to share some of my journey and my story and my science with you all. So we're going to just jump right in. Just as an overview of what I plan to talk about, I'm going to give a little bit of background on just my career path and how I got here. And then I'll talk about a bit about my current thesis work, and then finish up with navigating pregnancy, which is what I'm currently doing. So we'll have a little science sandwich there. So just to start, how did I get here? Well, I was born and raised in Kansas City, Missouri. I am biracial, so I'm Black and white. I also recognize that I'm quite white passing. So I think it's important to just acknowledge the privilege that comes with that and how that has also impacted my journey as a Black woman navigating the world. So I think it's just, like I said, important to acknowledge that. Growing up, when I wasn't in school, I was doing gymnastics with my sisters. I was like pretty much my whole life. We spent 20 to 30 hours a week in the gym practicing. So that was definitely a big commitment, definitely informed my work ethic and my time management skills and things like that. But like I said, when I wasn't in the gym, I am still a big nerd. And actually how I got interested and introduced to neuroscience in the first place was this high school summer camp called the Missouri Scholars Academy. And so basically we got to go to the University of Missouri for a couple of weeks, and we had a major and a minor. And my minor was this class called the thinking brain. And so in it, we got to learn all these questions about how the brain works and the mind-body problem and all these unsolved mysteries about how we are, who we are. And at the end, we actually got to dissect a human brain. And that was just the coolest thing that I had ever done in my life. And I have been hooked ever since on studying the brain. And so with that, I decided to go to Washington University in St. Louis for college, where I majored. They have a unique major there that combines philosophy, neuroscience, and psychology. And the reason that I was able to go there was actually because I had a full ride scholarship through the John B. Ervin Scholars Program, which is a phenomenal scholars program, a phenomenal community. If there are any high schoolers in the audience thinking about applying to college, I would definitely recommend looking into this because it's just an incredible experience and group of people to get to spend college with. Also during college, I got my first introduction into research. I had never really heard of research as a career path. Like I knew I loved asking and thinking about questions about the brain. And as I got to college, I took this freshman seminar called Researching Research. And they were like, research is when you get to ask questions as a career. I was like, great, perfect. That's what I want to do. And so every summer I participated in a different research experience at a different lab, just really kind of exploring what's out there and different lab types and different types of neuroscience. And then also during the school year, I'd spend that my junior and senior year in the lab of Dr. Robert Giroux studying pain processing. And through all of these experiences, you know, even when things weren't working or we got unexpected results, I was just loving the experience. And so I was like, all right, this is definitely what I want to do with my life. I want to study the brain. I want to ask questions. And so with that in mind, I learned that to have a career asking questions, I needed to go to grad school. And so I applied to several different schools and ended up choosing Vanderbilt University. I joined the lab of Dr. Danny Winder, and the lab's focus is really on this intersection of stress processing and addiction. And while I've been here, I've tried to be very productive. I've been a co-author on multiple papers, and I've applied to a lot of grants. I would highly, highly recommend applying to all the grants that you can. You won't get any grants that you don't apply for. And there were a lot of times when I was applying, I was like, there's no way I'm going to get this. And then I got them. So I'm happy to speak with people more if you have questions about any of these, or if you want to talk offline, you know, I'm happy to talk more about the grant writing process. But I think it's just super important to just practice writing as many grants as you can. So that's kind of just like this linear look at my trajectory. It looks very like professional and straightforward when you put it together like this. But if you zoom in, there were also a lot of personal experiences that informed my career path. So growing up, my dad struggled with drug and alcohol addiction, and my parents were divorced. There was a lot of pressure as the oldest child in a single parent home to like be the rock and like support my sisters with their homework and support my mom and all of these things. I took that job very seriously. So that informed a lot of how I navigate the world. In college, I was one of very few Pell eligible students at WashU. WashU has actually come under fire for being like one of the lowest concentrations of Pell eligible students in the nation. I was also in St. Louis at the time of Mike Brown's murder. And so that definitely informed my views on dismantling systems of oppression and wanting to do that in all spaces, but definitely academic spaces as well. My mom developed an opioid addiction that I didn't know about, and I had been sending money home from my research fellowships that I thought was helping pay the bills. It was actually just fueling her addiction. Two people closest to me, my boyfriend at the time, now husband, and my best friend had their first experiences with major depression and suicidal ideation. So it kind of, between all of this and like not seeing the signs and not knowing how to help the people closest to me, that definitely instilled a very deep-seated passion for addiction and mental health research by the time I got to grad school. And so here I am, very motivated to study addiction, also very passionate about outreach and teaching and just like letting people know about what addiction is and about mental health and improving spaces for underrepresented individuals. So yeah, I just mentioned all of this to highlight the important but often overlooked role that personal experiences can play in shaping our professional lives. So now I spent all this time talking about how I want to study addiction, but my title says I'll be talking about stress. So, okay, what's the relationship there? Well, as many of you probably know, addiction is a chronic cycling disease that cycles between these phases of use and then withdrawal and then craving and then cycles back into relapse of the drug use. And stress is actually one of the top reported causes of relapse in patients. And then in rodent models, stress can also reliably cause reinstatement, which is just the word that we use for relapse basically in rodent models, across drug classes. You can train a mouse or a rat to take a drug, and then you can train them to stop taking a drug, but if you expose them to stress, they'll go back to taking that drug. So also as I transition kind of more into the science piece of it, I do want to reiterate that, or highlight that, like, please ask any questions about anything. Asking questions is super important. something that I've had to actively work at, something that I still get very nervous about every time I raise my hand in a seminar, but just like the more practice that you get, the better. So please ask questions, even if they seem like simple ones. And all of this is fair game about my journey or about my science or about pregnancy later on. So just put all your questions in the chat. I'm happy to answer them. So with that, like I said, stress is a key driver of relapse in many cases. And a key brain region involved in this is a region called the BNST, or bed nucleus of the stria terminalis. Now, the BNST is really this node that integrates a ton of information from a ton of different regions, and it's important for things like emotional state and mood and fear and anxiety, and has been shown to really play a key role at the intersection of stress processing and relapse. In addition to integrating information from a lot of regions, it itself is an extraordinarily heterogeneous brain region. There are a ton of different cell types that can be classified by their expression of various proteins and peptides. And so this is just a small subset of the ways that these cells can be classified. One of the most heavily studied are these cells that express corticotropin releasing factor, or CRF, and they've been known to drive anxiety-like behaviors. They can drive relapse if you activate them. And so they were kind of looked at as like this key to this stress response and to this interaction with addiction. But unfortunately, with a lot of clinical trials, they really haven't had success targeting this CRF system. And so I think it's really important to look at how all these other cells are interacting. And we have a lot of work to do to understand this complex brain region. There's been a lot of work trying to characterize these. But there's, like I said, a lot of work to do. And the cells that I'm especially interested in are these cells that express this protein kinase C delta, or PKC delta. And so the rest of my talk is actually going to be focusing on this cell type specifically. That's what the pretty much entirety of my thesis work has focused on. And when I started my thesis work, there was actually no studies on what PKC delta cells in the BNST did or what their role was. As I've been in grad school, there's recently been some work implicating them in feeding behavior. And then in a related brain region, the central nucleus of the amygdala, there's also a population of PKC delta cells there, and they've been implicated in things like fear responses and stress responses and in addiction and in feeding. And so my work, my thesis work has really been focused on, okay, what role do these BNST PKC delta cells play in stress responses and anxiety-like behavior? That's what this region is known for. How is this cell population in particular contributing to that? And so the first question that I wanted to ask was figuring out, okay, what does the activity of this cell population look like during a stressor? And so in order to do this, to answer this question, I use a technique called Fibrophotometry in conjunction with this molecule called GCAMP. And so GCAMP is this modified calcium sensor that in the presence of calcium will fluoresce green. And so when neurons fire action potentials, there's this influx of calcium that happens. And so you can use this as a proxy of neuronal activity by detecting fluorescence when there's this increase in calcium, with the assumption that that is an indicator of increased neuronal activity. And so what I was able to do was to express this GCAMP molecule specifically in these PKC delta cells in the BNST. So this is just showing an example of, it's a very anatomically specific restricted population, which is another just interesting piece about these cells. But I'm able to express GCAMP specifically in this population. And then you can implant a fiber optic implant down into the brain and detect the fluorescence in awake behaving mice as they're going about their day, as they're doing different activities, and investigate, okay, what does the real-time activity look like of this population as they're doing different tasks. And so the first task that I looked at was this novelty suppressed feeding test, or NSFT. And what this task involves is placing the mouse in a large open box, and you put a food pellet in the center. And now the mice have been food deprived, and so they're motivated to go take a bite of that to go take a bite of that food pellet. But mice are creatures of prey. They don't particularly like to be in bright open spaces. And so there's this conflict that's elicited, this approach avoidance conflict, where they want to eat the food, but they don't necessarily want to be out in this exposed area. And so what I can do is measure the activity of PKC delta cells every time they approach this food pellet, which they actually approach it several times before they'll actually take that first bite. So I can look at those approaches, and then also that last approach where they actually finally do take that first bite. And so just walk through, I'll show a couple of graphs like this on the x-axis here. We have this timeline to the food approach, where 0 is when they enter that food zone, or in this case, when they enter that food zone and take that first bite. And then this is the measure of fluorescence, or the proxy of activity in PKC delta cells. And so what I found was that when they enter this food zone, then we see this increase in activity of PKC delta cells that is even more present in that final food approach, when they actually overcome that anxiety and take that first bite. So that was really interesting, and it hinted that, okay, these PKC delta cells are playing a role in this like stress processing, decision-making behaviors. But like I mentioned earlier, PKC delta cells have been implicated in feeding in general. And so I also wanted to conduct some studies where I was looking at stress processing, but dissociated from the context of food. And so in order to do this, I used restraint stress. And typically this would involve placing a mouse into a 50 ml conical tube for an hour, which I think would be stressful for anybody. And so this was how the lab had previously been doing restraint stress, but this is really not compatible with the cable that's required for our photometry system. And so when I had first joined the lab, I had seen people doing restraint stress. I had done some restraint stress, and the lab was just starting up with fibrophotometry. And I remember mentioning to some of the senior grad students and postdocs, I was like, wouldn't it be so cool if we could do fibrophotometry during restraint stress and record like during that activity? And they were like, well, yeah, but we can't do it with the tubes, and we don't want to just like cut a hole because you have to hook them up ahead of time to get a baseline. So, if you can figure out a way to make this work, then go for it. So I was like, okay. I was new in the lab. I was still waiting for like mouse science to be established. I was like, all right, challenge accepted. And so through a very iterative process, I ended up designing this restraint device, recording signal transients accessible in a tube. And this was honestly probably one of my favorite things that I've done in all of grad school, was just starting with literally 50 ml conical tubes and like scissors, and trying to cut and hack my way into this idea of how to make this work, and ending up with this final polished product that has allowed us to now conduct restraint stress while recording live activity from neurons. And so I mentioned this just to say, especially for people who are more junior in their career path, like if there's something a question that you need to answer and you don't have the tool, and try to make it if you can. So anyway, we had this device, and then we actually paired this with a software called DeepLabCut. It was really pioneered, the use of it and application of it with restraint stress was pioneered by a grad student, Joe Lusiger, in the lab, former grad student. And so what this allows us to do is track different positions on the mouse. And so we can track things like tail movements and head movements, and then whole body movements as they're struggling in this restraint device. So throughout restraint, mice will go through these periods of relative inactivity, and then these periods of active stress coping when they're like actively trying to escape. And so we're able to then align those active bouts with the fibrophotometry signal and see what happens when they're actively dealing with stress. And so when I looked at PKC delta cells, I saw that once again, when they're doing these whole body struggle bouts or stress coping behaviors, we again see this increase in activity of the PKC delta cells. And so with that answered the question, okay, during experiences and exposure to stress, these PKC delta cells are being activated. So then the next question was, okay, what happens if we go the other way? If I drive activity in PKC delta cells, what does that do to stress and anxiety-like behaviors? And so in order to do this, I took advantage of a tool called channel rhodopsin, which is a light-activated ion channel. And when you express it in populations of neurons and then shine light on those neurons, you can drive activity of that population of interest. And so the first question that I wanted to ask was just, is activating PKC delta cells aversive or rewarding? Do mice like it or do they not? And so in order to answer this question, I used a task called real-time place preference, where you put the mouse in this 2-chamber box, and when they enter one side, you turn the light on and it's going to stimulate PKC delta cells. And on the other side, you don't have the light on. And so then you can just see where mice prefer to spend more time. And if they prefer to spend time on the light side, that means that it's more of a pleasant experience. And if they avoid that side, then it's more of an aversive experience. And I also wanted to look at this in the context of stress. So if mice were in a stress state, does that impact the valence of activation of this population? And so what I found was that there weren't any differences whether they were stressed or not stressed, but in both cases, driving activation of PKC delta cells did result in this mild aversion of the side of stimulation. And so, okay, so mice don't particularly like to have their PKC delta cells activated. What's happening with anxiety-like behavior? And in order to do this, I use 3 different tasks that are, again, all relying on this idea that mice don't like to be in bright, open spaces. So you can look at things like the open field test, which is just a big open box, and see how much time they spend out in the center versus hugging the outer walls. And the more time that they spend in the center, the less anxiety that mouse would be displaying. I again use the novelty suppressed feeding test, except this time what I'm measuring is how long does it take for them to go and actually take that first bite, with more time indicating that they are more showing more anxiety-like behavior. And then the elevated plus maze has these open arms and these closed arms, and you look at the ratio of how long they're spending in each. Again, spending less time out on these open arms would indicate more anxiety-like behavior. And so when I drove PKC delta cells, we saw no differences in the open field, but for the NSFT test, we had a trend towards an increased latency to take that first bite, which is hinting at this slight increase in anxiety-like behavior. And for the elevated plus maze, we see a significant decrease in the time spent on the open arms, which again is consistent with this increase in anxiety-like behavior. So suggesting that stimulation of PKC delta cells is anxiogenic, which just means drives anxiety-like responses. And that answers that question. Okay, activating PKC delta cells is a little aversive and it can drive anxiety-like behaviors. So then the last question that I'm going to talk about today was trying to get, okay, so we see these like slight effects on the valence and on anxiety-like behavior, and they seem to be activated in these stress responses, but we really still don't know a ton about these cells. And so to get a better idea of what their function might be, we wanted to place them in their broader circuit context and understand, okay, where are these cells getting information from? Who are they listening to in the brain? And in order to do this and look at inputs to PKC delta cells, I use this technique called rabies tracing. I'm happy to go into more details about how it actually works if anyone has questions about it. But in general, it allows you to look at just single synapse away inputs to a specific population of interest. And so I could combine this and express this rabies virus in PKC delta cells to look at all of their inputs. And then I combined this with whole brain clearing, where you render the whole brain essentially transparent by removing all of the lipids, and then doing whole brain light sheet microscopy, which lets us image the entire brain at once without having to do serial sectioning, which can lead to, missed slices and cutting off things. And so it's this really, really, I think, awesome technique that lets you unbiasedly look at all of the inputs to the population across the whole brain. And so when we did this, this is an example of a mouse brain where you can see this is the BNST is right. Well, the dorsal BNST is right here. This is ventral BNST. And the PKC delta cell starter cell population is right here. And then all of these other cells are sending inputs to the PKC delta cells. And we can look across the whole brain here, where what you're looking at is a whole cleared brain, where this is the front, this is the back, the top's up here and the bottom's down here. And all of these white dots are cells that are projecting to the PKC delta cells in the BNST. And so when I look at this, what we found was that it seems like these cells are receiving a lot of input from regions involved in sensory processing, food consumption, and affect or emotional processing. The affect and the consumption were expected. Like I said, they've been implicated in feeding. The BNST is known to have connections with regions like the hypothalamus that's involved in regulating feeding, and also regions like the central amygdala, the rest of the extended amygdala, that are known to regulate emotions. These were expected. What was less expected was a lot of this sensory processing, because the BNST really hasn't been thought of as a region that receives a lot of sensory processing input. So it's pretty interesting that these cells in particular seem to really be receiving a lot of input from these regions. And so still, learning a lot about these cells. There's a lot of questions that I'm still in the process of investigating that I don't have time to get into. We have some really interesting preliminary data starting to look at outputs of PKC delta cells and starting to look at just their basal electrophysiological profile. And so I'm happy to get into any of that if anyone has questions. But for the sake of time, I'm gonna just kind of wrap up the science part by talking about just a couple other future directions. Like I said, I want to know what's going on, like what does their just general activity look like at baseline? And then also, how is that impacted by experiences and exposure to stress, especially any differences between acute versus chronic stress? It seems like this population might be particularly tuned to instances of chronic stressors and really salient exposures to stress. And then also interested in looking at, okay, what happens if we inhibit this cell population? What does that do to anxiety-like behaviors after exposure to acute or chronic stress? And then finally, I also am interested in what role PKC delta expression itself is playing within this population, because that really has not been investigated at all. What's unique about this population compared to most of the other cell types in the BNST is that this population is defined by the expression of a kinase, whereas all the other populations are defined by the expression of a neuropeptide that signals, that's released from neurons. It binds to neurotransmitters or binds to receptors. And so they have these like functions, but it really hasn't been looked at. Why is this kinase marking this population? So I'm in the process of doing some knockdown studies to investigate what role the actual expression of PKC delta is playing. But with that, a lot of this work is temporarily on pause because I am coming up on my due date. I am due to give birth on November 4th, thereabouts. So about 5 more weeks to go. Due dates are notoriously imprecise though. So that's like plus or minus 2 weeks really in either direction. And for the love of all that is good in this world, I really hope that it's on the minus side. But with that, I'm going to transition to talking about kind of my current experience with navigating pregnancy during grad school. And so I do want to preface this with the fact that every pregnancy is different, and many people have wonderful, magical pregnancies, and just really enjoy this experience. That has not been the case for me. Sorry, I just need to plug in my computer. Okay. I haven't had a great experience with pregnancy in general, and I've actually been pretty nervous to share about this. I kind of went back and forth about kind of how I wanted to frame this and what I wanted to talk about. So, there's already so much stigma around like female trainees and pregnancy and women in the workplace starting families, but ultimately decided, I really wish that I could have heard someone talk about an experience like this before going through it myself. So here I am giving the talk instead, and I hope that this is helpful to people. So I'm gonna dive right in. So I'm gonna just start with just a couple of things that went into my decision to go ahead and start a family during grad school. I want to recognize that not everyone or not every pregnancy is planned, but mine was probably hyper planned, which I'll get into. But these were just some of the things that if you're thinking about starting a family in grad school or at any time, some factors that you might want to keep in mind. So the first one was finances. And that's probably one of the biggest dings against starting a family in grad school is, compared to faculty positions or even postdoc, you're in a much less stable, secure financial position. Well, it's secure. It's just, you're not making as much money. But we figured, discussing with my husband, we decided that we could make it work. And then I also wanted to touch on a couple of newer resources that have been made available. The NIH recently added this $2,500 per year childcare supplement, which granted in the grand scheme of things does not go very far towards childcare, but it's certainly a step in the right direction, I think. And I believe that this will soon apply to T32 supported trainees as well. I want to take just a second to say, originally, for those of us in the F99 phase of the DSPAN, they said that this would also apply for us because our funding is essentially identical to that of the F31 recipients. And then they were like, psych, just kidding, and literally took it back. So in June, we got this notice that they were like, oh, you guys don't qualify anymore. You guys don't get this childcare support, which I find frustrating, in part because there's not that many of us. So it's honestly would not cost them very much at all. But if anyone wants to help me raise a big stink about this, I would appreciate any movement that we can get on getting this money back. I would love to have it. It would be very helpful. But anyway, finances should be taken into consideration when you're trying to think about family planning. Another thing is just external support. And so, a lot of us in grad school were not close to family. And so I think that's something that's important to keep in mind. This was another thing for my husband and I. We're we don't have any family in Nashville. We do have a lot of great friends. And so we think we have a supportive enough network and our family's close enough that they can get to us to help out. So that was important for us to consider. I hate saying biological clocks, but biological clocks are a thing to think about. And my husband and I, at least at the time we were discussing this, we're planning on having multiple children. And so it didn't really make sense to wait to start until I was tenured in a faculty position and already 35 or 40 if we wanted to have 3 or 4 kids. But the biggest one for me was this career timeline and flexibility aspect of grad school. So as I was thinking about this decision, I met with and spoke to a lot of women who had had children at various time points in their career trajectories. And I think one of the most useful pieces of advice or insight that I received was the fact that, as a faculty member, if you're if you just land a faculty job and you're trying to get tenure, you are on the clock. There's a lot of pressure to really have just like extraordinarily productive years, like right from jump, right out the gate. As a postdoc, you're also kind of on this like time crunch funding, like you're supposed to be very productive in this like short amount of time. But as a grad student, you actually have a lot of flexibility. Like no one cares really if I take 5 years versus 6 years in grad school. Like that will have pretty much no impact on my future career trajectory. And so that was one of the biggest points. It was like, okay, you know what? I think that this is something that I want to go ahead and do. And so my husband and I, we decided, all right, we want to start our family while I'm still here in grad school. So the next question was when? This was like an hours long discussion and like a very distilled version of it. And I recognize that this is kind of a weird way to decide a family planning trajectory, but this is the process that I went through. So I'll just share briefly my thoughts that went into deciding on when to start trying to have children. And so we started in July of 2020 when we sat down and just decided, okay, we're going to figure this out. And we decided, mostly me, to frame this around the Hannah Gray Fellowship, which is due in January or December of any given year. And so it has a time window of when you can apply, and it's only up to a certain number of months out from finishing your Ph.D. Generally, it's 16 months. This cycle is longer because of COVID, but generally it's 16 months. So I was like, okay, to have the best shot for this fellowship, I should try to graduate between September and December of any given year so that I can try to apply twice. And so took into consideration how long I thought it would take me to graduate, and also take into consideration, okay, if I'm trying to graduate and have a child, like what's the time frame that I think I can get this done in? And we settled on 2 years from July of 2020. So that put my anticipated graduation goal at September of 2022. And we were like, okay, we want to move with like a child who's around 1 year old because, you know, we want more kids. We don't want to have another child like right when I start postdoc, but also we don't want to move with a newborn. So we're going to start trying in February of 2021. I was like, awesome. Got the timeline, got the plan, ready to go. In the meantime, I was like, okay, I'm going to be productive in lab. Granted, it was still COVID, but I'm going to get stuff done. And I'm going to get all this research done and just like binge everything I can find about pregnancy and birth and parenting, because I knew nothing. I like have like no maternal instinct. I've never babysat a day in my life. I've never changed a diaper. I've fed a baby like twice. Like I had no experience with any of this. Like my husband is the one with like the instincts around parenting and like experience with small humans. So I was like, okay, I'm going to do all of this research. I'm happy to share my favorite resources with anyone who's interested. And then once I'm pregnant, you know, during that process, I'm going to crank out a ton of research, and then I'll go on maternity leave, and then I'll have this period of, you know, kind of normalizing back and figuring out navigating lab with a child, and then graduate in September of 22. What actually happened was, yes, I was productive in the time before pregnancy. I did learn a ton, and I thought I was completely ready and like knew everything to expect that pregnancy could throw at you. And then by God's grace, we were successful on the first try, which I know is not everyone's experience, but our plan was February of 2021, and that worked out. But pretty much from then on, it was just that's where the plan fell apart. So I, again, know that everyone's experience is different, and a lot of people are generally fine throughout their whole pregnancy. Some people even really enjoy pregnancy. I don't understand that perspective. That has not been my experience. And so I don't want this to just be me like ranting about pregnancy, but I also do want to keep it real and like true to my experience. So I'm gonna try not to just like spend too much time on this, but overall for me, pregnancy has ranged from like tolerable at best to actively terrible. The first trimester morning sickness for me was extraordinary nausea all day, every day for 8 weeks straight. It, you know, when I did have brief breaks from the nausea, I was just so tired and my brain was so foggy. Like it felt like my brain was just broken. And I remember trying to push through lab experiments that I had already started or had already planned. Like at one point I had this chronic stress experiment that I had planned where I had 5 days straight of restraint stress, and it was in this little side room in the lab, and I brought in 3 extra chairs, and I would start a mouse in restraint and then just lay down and pass out until it was time to switch mice. And this was just really not sustainable. So I looped my PI into kind of my experience of like, hey, this isn't working. He was very understanding, which I'm incredibly grateful for. And so I kind of was like, okay, I have to kind of step back. Like my mind and body are not in it. I don't want to be in the lab, making mistakes. And also just like, I just like couldn't push any further. So fortunately, the second trimester did bring some relief. The nausea passed. I had some energy back. I wouldn't say it was a pleasant time. Like I was still, it was still, I was just ranging from uncomfortable to actively in pain, like all the time. But I felt like my brain was working again. So I was like, okay, the worst of pregnancy is over. I can do this. We're going to revise the plan. And I came up with this folding plan of everything that I wanted to get done between then and my due date, keeping in mind, I was like, okay, well, due dates are imprecise. So finish everything 2 weeks before my due date. And this is the new plan. I was like, all right, we got this. Third trimester, all of the unpleasant symptoms kind of just ramp up to like a new extreme. And also at the same time, throughout all of this, in addition to like just generally having a terrible time, is you also have like all of these millions of things that you like have to get done to actually prepare for a child coming. And the nesting instinct is like very real and very powerful. That makes all of the baby related things seem so much more interesting and urgent than like everything else you have to do, regardless of like their actual relative importance. So I was trying to balance all of this. Third trimester, it was really not a pleasant time, but I was pushing through because after all, I had this plan, right? I have these experiments. I was like, okay, if I can just like make it through these experiments, it'll be fine. But for me, kind of the major blow or straw that broke the camel's back, if you will, was getting diagnosed with gestational diabetes. And I do actually want to spend just a little bit of time on this because it is something that gestational diabetes, there's a higher prevalence in Black women and actually pretty much every non-white population. And the maternal health care system is notoriously trash for Black women and letting a lot of Black women slip through the cracks. And so I'm happy to go into the whole saga if anyone's interested, but basically with this, I've been left to my own devices to like figure out how to navigate it. And so it's been extraordinarily challenging. And with it, like from bad advice and lack of advice, I had sent my body unbeknownst to me, like into this extreme carb withdrawal, which is bad for anyone, but especially bad in pregnancy, to the point where over the past couple of weeks, I've been so, my state has been so unpredictable that it makes it impossible to get work done. You know, one day I'll be literally laying on the couch with like absolutely no energy, like feeling like I can't even move and just like crying for hours. And then the next day I'm fine. And then the next day I might start fine and then fall back into this state. And it's just been like this whiplash of trying to navigate this and trying to manage my physical health and my mental health. And at the same time, you know, as I was struggling with all this, like I was supposed to be doing lab work. I had all of these things planned out that I was going to get done. And I tried to come in a couple of times. And like when I was in the lab, I felt like I could kind of just ignore my body and get through some stuff. But then I'd get home and just crash. And it was not sustainable. And it was not healthy. I was losing weight. You're not supposed to lose weight in the third trimester of pregnancy. And so basically, I had to be okay with letting things go. I had to cancel pretty much all of my planned experiments that I had for the remaining weeks of pregnancy. Because like I said, I never knew how I was going to be on any given day. So I couldn't like plan ahead of like, oh, tomorrow I'm going to get this done, because like, I didn't know what tomorrow would be like. And so I had to stop, and I looped my PI in, and, you know, I let him know that like I needed to change the plan. I have plenty of data analysis. I can always read papers from home. There are lots of things that I can do that aren't on strict time schedules. And so I am, I just, you know, I had to tell him, I was like, I can't do this. And again, thankfully I have an amazing, amazing mentor who was like, that's fine. Like, take care of yourself. And so I had his support, but internally, like for myself, even though I was saying, I was like, okay, you know, I can't do this. Like, I need to take care of myself. I felt like an absolute failure of a scientist. I was like, everyone else that I've seen in pregnancy seems to be able to just like push through. Like, you know, the narrative is that you push through until you're like in labor and then the struggle starts when the baby gets here. So I was like, what is wrong with me that I can't do this? But that was where my body and my mind were at. And so I've been learning to just do what I can when I can. And really like focusing on just like taking advantage of when I feel good, I'll get things done. And when I don't feel good, I'm going to take care of my body, in part because like, you know, my health is worth it. And also it's not just my health. Like, it's my physical state and emotional state are intricately and inextricably tied to the development of this growing human. And so, you know, it's definitely been a process. I'm still working on it. It's still something that I'm struggling with. Again, like I'm still in it. So I don't like have this like long-term perspective. But I'm working on it. And I think it's a really important thing to keep in mind. So I just want to kind of finish up with some lessons that I learned of how to make pregnancy work. And like I said, you know, every pregnancy is different. This is just my experience. And you might not have these struggles, but you might. And there's so many different things that can pop up during pregnancy that I think it's just worth having a plan and like being ready. And so the first thing that I would say is to gather your support team early and ask for help when you need it. Have someone in lab who is gonna be understanding. For me, that's been my PI. But if you don't have a great PI or a great relationship with your PI, then have someone who can stand up for you and be like, look, like, take a break. Having someone who can like stand up for you and be like, now is the time to prioritize you, has been so important. As someone who's in a position of power, right? Who can say that with, I don't know, with authority, has been really useful. If you can, I didn't have this, but I think it's something that would have been really helpful. Have a set of helping hands that can jump in if things come up. things might not come up. But if you can have like an undergrad or a lab tech ahead of time that you can train, who can jump in, I think that that would have been something that was really, really helpful. And then therapy, highly recommend therapy. Therapy is great. My therapist has been amazing through this whole process. Secondly, plan what you can, but flexibility is absolutely the key. Generally speaking, the second trimester will probably be the best. So if there are things that you like, know you like have to get done, like try to align those with the second trimester if you can. But again, there's no guarantee. And so like have your ideal plan, but be ready for it to not happen and be ready to just do what you can when you can. Now is not the time to overcommit. You have to be a master of prioritizing what tasks are most important and like absolutely have to get done and what tasks don't. And the level to which those tasks need to be done. For example, you know, I was putting together this talk and I was trying to navigate like this catch-22 of I want this to be a really good polished talk and I really want to get this message across, but also it's stressing me out and I'm exhausted and it feels a little hypocritical to stress myself out over a talk about not stressing yourself out. So, you know, I typically have very, very high standards for myself. And is this like as polished as I maybe would have liked? Probably not, but I hope that it still gets the message across. And then finally, listen to your body, give yourself grace because growing a human is really, really hard. And my PI recently gave me some like remarkably simple, but like very profound advice in just reminding me that like this is temporary and that the lab will still be there when I'm ready to get back to it. And in the big picture of like my career timeline, if I plan to spend decades in research, like this is a very short time frame. And so were some of my research plans put on hold? Yes, but my career plans are still very much intact. So with that, I know that I've run a little long, so we don't have a ton of time for questions, but I just want to thank my whole lab, thank my funding sources, and go ahead and open it up for questions. So, yeah.
Thank you so much, Kelly, for that amazing talk. It was great to hear about, you know, your research on stress and addiction, and then also your experience being a expecting mother as a graduate student. I feel like we all hear a lot about, you know, how having a child during different points in your career can affect women in particular. And it's so kind of you to open up and share with all of us about your experience. So we have a few questions. So I'll start with some questions about your research. So there's a question from Teresa asking, complimenting you on your talk and your very interesting review on your work, and asking, do you think that these insights from your research will eventually inform treatment for trauma as a special case for stress? So like, do you think that this research will inform stress treatments?
I think that there's definitely a possibility of that. What I think is unique, especially about the cell population I've been looking at, the PKC delta cells, is it doesn't seem to be this like sledgehammer effect on stress processing. It seems to be more of like this fine-tuned like dial. And so I think that that may be really important in thinking about like treatments where if you just like hit this like major system that's like broadly throughout the brain, then maybe that's why it's not working because it's involved in so many other different things. But if you get something that's like more specifically fine-tuned for what you're trying to treat, then maybe that's going to be more effective. So, I mean, I hope so.
Yeah, that makes complete sense. And it seems like a lot of the treatments that we have now are more like sledgehammer models. And so we have another question. Do you think that this work may also provide insight into why some people become addicted while other people who maybe try drugs don't?
So I think that's a really great question. It's definitely a question that I'm interested in. And I think that understanding differences in, you know, how cell populations respond to stress can definitely provide insight in that. Unfortunately, in my thesis work, I don't think I'll probably get to actually answering that question in particular, but just like knowing, so maybe some people's cells are hyper responsive to stress or take less stimulation or less activation to be in this state that is primed for relapse. So I mean, I think that with definitely more work, but I think that question could be answered. I probably won't get to answer, but I think it's a really important one, and it's one that I'd love to look at long term for sure in like my future career.
Yeah, it sounds like you're laying the foundation for future work for other people and yourself to build on, which is awesome. So we have another question asking more about your family history. So it's by an anonymous attendee. How have you navigated supporting your loved ones through substance abuse disorder and mental health conditions while also taking care of yourself?
That's a great question. It has not been easy. I mean, so growing up, like with my dad, I was a child at the time, so like that didn't really fall on me, and I was kind of protected by my mom from a lot of the like particularities of that. But with my mom, I think that was particularly devastating because it was like this happened when I had gone away to college, and so it was like I had left behind my family, and like I wasn't there to see the signs. So it's been something where I try to use like my experience and perspective of like, this is what I study, like this is real like science, like this isn't just something that is wrong with you or like just like a moral failure, and like really try to like encourage, I guess, my loved ones with the fact that like, hey, like this is like what you're dealing with is real and it's really hard, and we're just trying to, like my whole career is dedicated to like trying to figure out how to help, but like you're not weak because of what you're going through. You're not like there's nothing wrong with you as a person. Like it's just biology. It's just like any other disease. And then in particular, like with my husband, trying to make sure that I take time for myself. I take time for ourselves. He's been doing a lot better. So, and my parents also. So it's, a lot of up and down. It's definitely really challenging, but I think making sure that I still make time for just me to recharge has been really, really important. Doing things that just I like to do, and that I can kind of step away from lab, and I can step away from family, and I can step away from everything else and just like recharge my batteries so that then I have more to give other people.
Yeah, and I think what you're saying and what you said in your talk is super important. Just, like demystifying mental health as like something is wrong with you, and like talking about it like a disease, like anything else that would affect your physical body. So that's super important. And so although you have a lot of questions here in the chat and in the Q&A, we only have time for one more. And so this question is from Jessica, and she says, they say, "Hi, Kelly. I'm currently in my second trimester and have also been struggling with feeling failure or for not being able to work as hard, but trying to give themselves grace. And they're just curious how you plan on planning your maternity leave, negotiating time to come back, and flexible schedules. And if you have any advice on that for others."
Yeah, absolutely. It's something that I've been thinking about a lot, especially because, I've had to ramp down my like experimental plans a lot earlier than I thought I was going to. And so now I feel like, oh, now I need to like get back to it as soon as I possibly can. But what have been trying to internalize, and I hope by externalizing it, maybe it'll help me internalize it a little bit more, is I can't plan everything. I can't know ahead of time like what my maternity leave experience is going to be like. I can't know for sure like what I'll be able to do on any given day. And so I think that the approach that has been most helpful for me is thinking about it like, okay, I'm going to make this whole list of goals, right? And so I'll have this comprehensive, like, these are the things that I would like to get done. And this is kind of like the priorities, or like the order of priorities, like what's most important, what's least important, et cetera. And then rather than like, okay, I'm going to get this done at this time and this done at this time and this done at this time, thinking about it more of like, okay, today I can work on this thing. Today I can work on that thing. Today I can work on this other thing. Today I can't work on anything. So like just trying to take it more as like, rather than having this like specific set out, laid out timeline of like, this is exactly when I'm going to come back and this is exactly what I'm going to get done. Like, this is my broad big picture goal and I'm going to get done what I can when I can. So happy to chat more, or just like have a commiserating session. We can just cry together if you'd like. Feel free to contact me. Always down for a good cry. So, yeah.
Thanks so much, Kelly. We were wondering if you had like maybe 5 more minutes that we could take of your time. Great, because we have so many questions and there's still a decent amount of people here. So we want to just give you the opportunity and give the attendees an opportunity to get more information from you and just hear more about your experiences.
Yeah. Also, I'm happy to share my email too if people want to contact afterward, but like I'm free to continue. So.
Okay, great. Thank you so much. Cut us off whenever you are ready, but we'll probably be another like 5 or 10 minutes if that's okay.
Yes.
So I'll post some science questions now. Someone asks, in general, what does local connectivity look like locally for PKD delta circuits? As in PTK delta to PTK delta, PTK delta to local inhibitory neurons, things of that sort.
Mm-hmm. Yeah, no, that's a great question. It isn't something that has been well dissected. Based on some of the other literature, there is a lot of like intra-BNST connectivity of PKC delta cells that project to like the ventral BNST. That's been definitely like very clearly shown. And it looks like, based on my rabies tracing, that they are also receiving, inputs from intra-BNST sources, but it really hasn't been like well worked out, like what the ratio of interneurons versus like projection cells are. I am really anxious to look at some of the output data. I have brains that have been cleared to look at outputs, and it was one of the things I had to put on hold, so I don't know where they project to. But it does look like they receive at least a subset of input from the BNST itself. And the BNST is almost entirely GABAergic, so mostly inhibitory interneuron sources. So that definitely plays a role in their function and who they're listening to. But I don't know if I fully answered that, but I guess the full answer is we don't know for sure, but it seems like it's a mix of both. So.
Thank you. And so we have another question. It says, with the emerging increasing evidence of gut microbiota and its metabolomic bidirectional communication with the brain, do you think modulating gut microbiota could influence the expression of PKCD expression in the brain?
I mean, I definitely think so. It's not something that I've looked at all, or that I think anyone has looked at in the BNST. But I certainly think that it's a possibility. The gut microbiome is so interesting to me, and I think there's so much more work to be done. But I don't have an answer for that question for sure. But if I had to guess, I would say yes. So.
It sounds like another follow-up experiment for you to do later on. As if you don't have enough of those. Thank you. And so we have some more questions. This is more of a broad question about your scientific journey. Do you have a favorite project that you've worked on so far in your graduate program?
Well, I mean, the restraint device, which wasn't necessarily a project, it was like more of like I guess it was a project. It was like an engineering project. That was probably my favorite. You know, I've had kind of an interesting experience with like projects because pretty much all of my work has been centered around these PKC delta cells, and then I had another piece of it that kind of fell off because the tools weren't working. But like really, it's just been like different aspects of understanding this like one population. I've been like very focused on that, which I wouldn't necessarily recommend because I think it's good to have other side projects. But I think for me, it's been like not one thing, but just like all of the small hints that keep popping up about like, oh, okay, this data over here is like suggesting this one function, but it like contradicts with this other data that I have. And so it's just been this like constant process of like, what are these cells doing? That's been very, I mean, frustrating at times as any project can be, but like it's always constantly like there's more and more questions. Like I have so many more questions now than when I started. So I think it's just like been the intersection of like all the different pieces and trying to fit them together. That's been my favorite part.
All right, Kelly. Well, thank you so much for your time. We appreciate you sharing your research and all of your personal experiences throughout grad school, your upbringing, and throughout your pregnancy. We all wish you a pleasant finish to your pregnancy. And thank you so much. And as Kelly said, you can contact her at her Twitter, which is right here on the screen. We will, the few people that sent us their emails, we will pass those on to Kelly so she can contact you personally. Thank you all so much for joining us. And thank you so much, Kelly, for sharing your experiences and your science.
Thank you for the opportunity. So I appreciate it.
Have a good day, everyone. Thanks, Kelly. That was so amazing.
Thank you. Thank you. Thank you. Oh,
so
all right.
Great. Oh, wow. So yeah, we'll reach out to you about compensation yeah, good luck with your pregnancy. We'll be in touch. I will personally be in touch to talk to you more about grant things. Thank you so much. And yeah, so anyone who asked about the recording, Brielle just put it in the chat, but it's available on our YouTube channel. And yeah, thank you so much.
Yeah. All right. Well, thanks.
All right. Have a good day.
You too.
Bye.
I could stop sharing my screen, but.
Oh, yeah.
Thank you so much, Kelly. That was wonderful.
Thanks for having me.
Yeah.
So.
Yeah, I hope you get a chance to I don't know if you have to run, but if you want to look through the chat, we save the people that ask for your emails, but just it's all people oh my gosh, this is such a great discussion. I'm so thankful, so wonderful, so inspiring. So I hope you get to look at it because people just loved it.
Oh, wow. Yeah.
Yeah.
Oh my goodness.
People were raving. It was so good.
It was so good. Yeah. And I just loved your candor.
Ashley, it's still live on YouTube.